It's my pleasure to introduce the next speaker, Dr. Chris Garcia. Chris is professor of molecular and cellular physiology and structural biology at the Stanford University School of Medicine. Chris is a member of Stanford's Wu-Tsai Neuroscience Institute, the Stanford Cancer Institute, and the Stanford BioX program. He's an investigator of Howard Hughes Medical Institute. He's a member of the National Academy of Sciences and the National Academy of Medicine. Chris's work investigates structural and functional aspects of cell surface receptor recognition and activation in receptor-ligand systems. And his talk today is Modulating Cell Physiology Through Ligand-Receptor Engineering. Please join me in welcoming Chris Garcia.
Thanks, Ken. Okay, is this on? Can you hear me? All right, what, now? Yeah, now you're getting it. Okay. So, yeah, it's great to be here. Thank you for inviting me. I remember cycling with Tim in Monterey quite a while ago, and Tim sort of talked about the initial, kind of vision that he had for this thing. It's amazing to see what it's come to and where it's going. And so I'm excited to be here because, this is what I think about all the time. I'm a cell surface guy, and I'm interested in concepts that you've heard about today. Ligand-receptor activation, how ligands activate cell surface receptors. And really, to me, it's about exploiting structural and mechanistic information using protein engineering to probe mechanism, but also to make molecules that could potentially have a therapeutic use. And so for me, it's about actionable value. And, I kind of have a reputation as a structural biologist, but really, the only reason I got into structure was because I wanted blueprints to do protein engineering. And so to me, protein structures are kind of like, I look at them sort of like a contractor looks at a house they're going to remodel, looking for opportunities to improve things and change things. And so when I decided to put together a talk today that really goes through some of the systems we've worked on in my lab over the years, from the immune system to the Wnt system to new ideas in induced proximity, so I'm going to go across multiple fields, giving snapshots of different examples of how we're implementing this type of cycle in my lab. So I'm going to start off with Wnt, which probably needs no introduction. I came into Wnt because it was this really interesting structural challenge of a lipidated growth factor that bound to a receptor, frizzled, that was a 7-TM receptor that had analogies to a GPCR, but it didn't really work like a GPCR. And so we initially started off by solving the structure, the crystal structure of the Wnt frizzled CRD complex, and extended that to the full-length frizzled structure by cryo-EM more recently. And then the most recently, we managed to finally trap, using protein engineering to enhance the affinity of Wnt for LRP, we managed to trap the ternary complex structure, which has now allowed us to sort of start to piece together what the whole signaling complex looks like at the cell membrane. And I'm not going to really talk about any of the biological implications here, and mainly focus on the way we really got into the engineering here was that Wnt is lipidated. It has an acyl group that is actually one of the major receptor binding determinants on frizzled. And when I saw that, I thought, this molecule, there's no way that it's ever going to have drug-like properties. We're going to have to figure a workaround here if Wnt is ever going to be a drug for regenerative medicine. And so, what sort of taught away from that idea was that frizzled looks like a GPCR, and GPCRs, through, as you know, the work of Andrew Cruz and Brian Kabilka and others, works through conformational change mechanisms, but it also had a co-receptor. So we tried the idea of replacing frizzled with a small bispecific antibody type molecule to heterodimerize it, and we managed to phenocopy Wnt signaling with the surrogate Wnt agonist. and really showing that all that's required for Wnt signaling is a heterodimerization. And these molecules, the first generation of these molecules, were active in every way Wnt was. They were systemically active in the liver, you can see. They had better biodistribution from Wnt, which really doesn't biodistribute very well, obviously, because it's lipidated, and it's particularly potent in bone growth and in the GI in activating stem cells. The problem with this molecule is that it was pan-reactive with all the frizzles. There are 10 frizzles, and they're expressed, different frizzles are expressed in different tissues to different degrees. So if you really want to use a Wnt surrogate agonist as a drug, you're going to need some type of tissue specificity. So we wanted to make molecules that could narrow the specificity to individual frizzled subtypes. And for that, David Baker and I, who collaborated on many different things over the years, we got together to design and engineer a repeat type molecule, not identical to the DARPins that Andreas invented, but similar, and designed it, and this is the structure now bound to the frizzled extracellular domain. The structure was exactly as the design, and what we did through sort of iterative cycles of engineering and design was to create versions that were able to seek out the differences in the different frizzled subtypes, and in some cases those are as little as single amino acid mutations in the whole protein. And we managed to get highly specific versions to different frizzled subgroups. And so these now turned out to have some interesting properties. So for example, in the GI tract, which mainly expresses frizzled 5 and 8, our frizzled 4 was inactive, the 127 was inactive, the frizzled 5 and 8 was active. Liver is mainly 1, 2, 4, and 7, and you can see our frizzled 4 and 1, 2, 7 binders are active here, whereas the 5 and 8 isn't. And they've also been useful for the organoid community. Hans Clavers has replaced Wnt in all their organoid work with our surrogate agonists, and we actually get much better quality organoids things than with Wnt. And so, these have taken on a life of their own. The Wnt surrogate agonists are now in clinical trials for healing GI disorders, also liver regeneration, and we're continuing to sort of fiddle around with this axis in the lab. Now, one other interesting part of this pathway is that frizzled has its own degrader molecule, the cell surface E3 ubiquitin ligase, which what it does is it degrades frizzled and takes it off the cell membrane, making cells unresponsive or very weakly responsive to Wnt. And what happens is there's a molecule called R-spondin, which binds to this ubiquitin ligase, and Yvonne Jones has done some structural work on this. And basically, it works by distracting the ubiquitin ligase away from frizzled and increasing the frizzled expression level. So what we did was we sort of took that natural example of R-spondin, and we've created molecules that can ligate E3 ubiquitin ligases to any target receptor and degrade it or lower its expression levels off the cell membrane. And we call those degraders, and other people like Jim Wells and others have also published papers in this space. This is showing you an example of what this looks like. We targeted this E3 ubiquitin ligase to EGFR. You can see it can take it off the cell membrane by as much as 20% or so, and it'll inhibit proliferation of these EGFR positive cells as well as cetuximab. So this is kind of a new field, the degrader field, and one of the limitations is you don't get complete takedown off the cell surface. So that's one of the questions is this enough to be therapeutically useful? And we're certainly going to find out. There are a lot of companies out there now that have started around degraders. So Wnt, the sort of the bookend for Wnt for me is the Notch system in developmental biology, and so we did some structural work in my lab several years ago on Notch jagged and Notch delta complexes, and this is the structure of how Notch engages jagged. It has some interesting structural features. One of them is that this idea that Notch is glycosylated, and it's one of the ways that it regulates its affinity for its ligands by this developmentally controlled glycosylation. And these glycans actually They sort of act as surrogate amino acids in the binding site to tune the affinity for different ligands. But what was the interesting mechanistic insight here was that we worked with the biophysicist Cheng Zhu to do some measurements looking at the mechanical force properties of receptor activation, and also we saw that in the complex the jagged molecule was bent compared to the unliganded structure, and we found using this biomolecular force probe measure what Tim Springer initially for selectins termed a flex bond, which we call a catch bond, which is that there's an increase in bond lifetime with applied force. So it's kind of this transient resistive force as this unbinds while it lets go. And this happens under shear force. And it's the way Notch uses to potentiate its affinity for its ligands, which are really almost unmeasurably low affinity. So under the conditions of shear flow in between tissues, Notch will be bent into this catch bond angle and engage Notch. And so what we did was we engineered a high affinity version of jagged and delta. This is a Notch activation assay. And you can see here's the wild type delta, and here's these affinity matured versions. And what was interesting was that these versions, we didn't touch the interface with Notch. We just locked that bent conformation in. And by locking in the bent conformation, we froze out the catch bond. So now it's a slip bond. That's the natural one that can bend. This is the one that's rigid. And so, we're still, I think mechanical force, like engineering mechanical force, is one of the great frontiers of protein engineering. We still don't know how to do this well yet. And this is kind of where we're at. The problem with these engineered ligands is they have to be anchored. They have to pull on Notch on some sort of anchored surface. So what we're trying to do now is we're focusing on this mechanosensing domain of Notch, which Steve Blacklow really has shown is the smoking gun for how Notch is activated down here. And we're trying to open this up using some sort of protein engineering strategy. And one is we collaborated with Andreas Plukven, who isolated these DARPins, which bind in 2 sites on this NRR domain. And what our goal now here is to see if we can twist or torque these 2 DARPins and open up this NRR to expose this cis-aryl bond for the gamma secretase and actually have a soluble agonist of a mechanoreceptor. And if we can pull that off, I think that's going to be a big deal because there are a lot of mechanoreceptors that right now you have to activate with anchored ligands. We're not there yet, but I just wanted to tell you about what we're trying to do. Okay, and so, we're at the point now where we can start going back to the basic biology and asking things like interrogating Wnt-Notch interplay, because now we have reagents, biochemically pure recombinant reagents that we can turn Wnt on or off and Notch on or off and really, start to go back to some of those original basic questions in developmental biology. And we're just at the beginning to do this now that we have the tools. Okay, I want to segue from here to the T cell receptor, which is a system I have a long-standing interest in, and this is the structure of a T cell receptor recognizing a peptide MHC, and so this interface is extremely interesting because it's got these variable loops in the T cell receptor. It's got a variability in the antigenic peptide. It's bracketed by these helices. And I've been basically obsessed with this recognition event for about 25 years or so, how the TCR is seeing this peptide. And so we developed a tool, again, a protein engineering tool, to really ask this question. And it's a yeast display system where we're able to display, billions of peptides presented by MHC on yeast. Steve Elledge has developed similar systems, and we're able to then screen these against T cell receptors and identify sometimes endogenous ligands, surrogate peptides, profile their cross-reactivity. And we found something very interesting out of this that was a mechanistic insight that was unexpected, which is that the affinity of the T cell receptor for peptide MHC can be decoupled from its signaling strength. In other words, we isolated peptides that had high affinity for the TCR but did not signal at all, as well as weakly binding peptides that signaled very strongly. And in the end, we narrowed it back down to the catch bond, which I just told you about with Notch. But this is a different kind of a catch bond. And I'm going to tell you a little bit about that story today. So to make a very long story short, we had two T cell receptors, one that I call the non-stimmer and one that I call the stimmer, recognizing an HIV peptide presented by this HLA. And structurally, they were essentially identical. There was nothing remarkable about the non-stimmer. They both, we could stain T cells, both their T cells with MHC tetramers equally well. Their affinities, as well as on and off rates, were firmly within the physiological regime that should activate these T cell receptors. But this TCR55 was not activated by the ligand in either IL-2 or CD69 or by any other metric, whereas 589 was. So what's the difference between these two things? And what we did was we took our yeast peptide MHC libraries and we screened against the non-stimmer TCR, and we recovered agonist peptides. There's the HIV peptide. These are now agonist peptide mutants that's now turning this T cell receptor on. And we collaborated with Brian Evevold, another person who does biomolecular force probe measurements. And what we found was the non-stimmer peptide was a slip bond, whereas the stimmer peptide was a catch bond, just like you saw with Notch. Except in this case, the catch bond is rather unusual. Rather than in a domain movement type of catch bond, it's between two relatively flat surfaces. And so, visuals really help me. So I sort of did this kind of, this sort of depiction here of how I think about catch bonds. They're these like little lightning bolts that occur between very few residues of the TCR and the MHC. And when you think about it, it's like this finger trap where, you put your fingers in there and you try and pull it and it catches. That's a crude, not very good analogy of a catch bond, but that's the way I think about it. And they are literally able, the chemistry of this interface is literally able to make the determination between turning signaling on and not able to activate signaling. So we decided to flip the question around. We were able to turn on the non-stimulatory TCR with the peptide. Is it a bidirectional event? Could we get catch bonds from the T cell receptor to the non-stimulatory peptide? So what we did, we developed a technique we call catch bond fishing, where we made very sparse libraries on the CDR loops of the T cell receptor. These libraries are on the order of thousands of total mutants. And we screened them against the non-stimulatory peptide MHC complex in a cell-based reporter system. And we selected for activated T cells that had low affinity binding, tetramer low. So binding low, activation high. And we got, and so here's an example up here. Now these are some of the clones we got. This is the wild type peptide. These are single point mutants in one residue of the TCR that you can dial up or down the activation potential. And all of these have acquired catch bonds, as opposed to the non-stimulatory TCR. And in fact, the catch bonds, the bond lifetime is proportional to the CD69 upregulation on the T cell. And when you measure the affinities, the 3D affinities of these mutants with the TCR, they're not in any way significantly different than the non-stimulatory peptide. So the catch bonds have the ability to decouple signal strength from the affinity of this interaction. And it's an engineerable parameter. So we tend to think about affinity being one of the few engineerable parameters between proteins. Catch bonds are a new engineerable parameter that don't depend on affinity. So the the real actionable value of this is in therapeutic TCRs and T-cell engineering and adoptive cell therapy. And the example we came to was this, T-cells now, TCR-T therapy largely depends on affinity maturation of T-cell receptors because of thymic education. Our T-cells tend to have blunted responses to tumor antigens, to self. And so many companies are now affinity maturing TCRs to tumor antigen. And here was one case of a TCR that went into the clinic against a cancer testis antigen, MAGE-A3, a good tumor antigen. And it was affinity matured, and they picked up a toxicity against a Titin peptide in the heart. It was an off target. And it was a big crisis because some patients expired, and so we went back to this T cell receptor. So this is now looking at T cell activation by the MAGE-A3 peptide. Here's the affinity matured peptide. It's activated very nicely. Here's the non-affinity matured peptide. So we started with the parent TCR and we catch bond engineered it instead of affinity maturing. And when we catch bond engineered it, most of the mutants had virtually no reactivity with the Titin, which is the cross-reactive peptide. No reactivity, even though they were roughly the same affinity. And their killing potential of the target compared to the high affinity TCR is the same or better in some instances. So we think this is now a way of generating highly sensitive and potent T cell receptors for immunotherapy that bypasses the risk of off-target cross-reactivity. So I want to move towards cytokines here, and that's another area I've really been interested for a long time since I was at Genentech as a postdoc. And we've managed to piece together the signaling pathway, in large part due to some recent advances by Caleb Glassman on structure of full-length JAK. And so we can now piece together a process whereby the cytokine binds with high affinity to a receptor associated with the Janus kinase. The second low affinity binding site receptor then forms a dimer with this subunit. It dimerizes the two JAKs via some domains. I won't get into the details today. And then the kinase domains underneath here, they undergo a conformational change. They transphosphorylate. They then phosphorylate the receptor, which recruits the STAT molecules. Once the STAT molecules are bound, they're phosphorylated, and then now they go to the nucleus. And this structure just presents a lot of opportunities for engineering and modulating cytokine biology. And really, for me, the muse here, so my neighbor at Stanford is Brian Kabilka, and I was watching his GPCR. GPCRs are like the ultimate tunable receptor. That's the whole GPCR medchem industry is based on bias and inverse agonists. It's a whole branch of pharmacology. And we actually determined the structure of a chemokine, which is a protein ligand bound to, sorry, this is a little offset here, bound to the GPCR with the G protein. And we engineered the chemokine to display the sort of pharmacological properties of biased agonism and partial agonism. So, we always wondered, could we do, sort of use the properties of GPCR pharmacology on cytokines and create these sort of, biased and partial agonists and biased signals. So we've done a lot of this, and I'm not going to go through all of it. I'm just going to give you some select examples because we have this, we have all these blueprints in the lab of all these houses we want to remodel here. And so now with the structures of these cytokines, we can now go in there and make mutations and look at the activities using these blueprints. So here's one example, IL-2, which is a system I've been also involved with for a long time. This is the structure we determined a long time ago. And what I just want to tell you about today is we made point mutations on the face of IL-2, which interacts with the second chain, which is the common gamma chain. And by doing so and slightly destabilizing that complex, we were able to essentially create this to tile the whole range of STAT5 activation from full agonism down to all these partials. And this is exactly what a GPCR partial agonist pharmacology curve looks like with a small molecule. And so what we did in the lab, and several people were part of this, I'm summarizing a lot of work, Caleb was part of this, we systematically profiled each one of these partial agonists in mice compared to wild-type IL-2 and asked, do they do anything different? And so one of them we call H9T, we worked with Warren Leonard and Nick Restifo. This is now looking at T-cell expansion by IL-2 and then H9T is the mutant. They expand just as well as IL-2, but it doesn't upregulate these exhaustion receptors. And it also has much stronger stem-like properties if you look at like the TCF1 and glycolysis. And in a adoptive cell therapy model that Nick Restifo's developed for a long time at NIH, when we precondition cells with H9T versus IL-2, we get much better tumor control and survival outcomes and T-cell persistence. And this is what the single point mutation in one of the receptor binding sites has this profound biological effect. Now that's H9T, which is a barely perceptible attenuation of STAT5. Another version, REH here, and again, this is Caleb's work. There's a theme here. Caleb was very prolific in the lab. This now selectively activates regulatory T cells over conventional T cells. And in some gene expression work, if we look at the T-cons versus the Tregs, we see that certain genes like BCL2 and MYC are strongly downregulated in T-cons, but virtually untouched in the Tregs. So there's a different hard wiring by these cells to the attenuated version of IL-2, and in a DSS model of intestinal inflammation, it activated Tregs and it had a protective effect. So really the point of this is how sensitive cells are to that signal strength of the cytokine. And you can really profoundly change the biology just by very small differences in the receptor interactions. As Yvonne alluded to earlier, the power of knowing the interface and being able to target it. Another cytokine that we've been working with is gamma interferon, sort of the other master regulator of the immune system. And gamma interferon is one of the most pleiotropic cytokines. Most immune cells have gamma interferons, and it's been really impossible to use as the therapeutic because it just does so many different things. And it's one of my other pet obsessions in the lab is like harnessing gamma interferon for immunotherapy. And the parameters that we're focused on is the fact that, gamma's primary role is upregulating class 1 MHC to drive up tumor antigen expression and make cells, more present viral antigens better and that sort of thing. But it also has this counterbalancing effect of upregulating PD-L1 expression. So there's kind of a neutralization of the immunostimulation by the PD-L1 expression. So Juan Mendoza in my lab determined the crystal structure of the gamma interferon receptor complex, and I'm not going to get into the structural details other than to just point out that there are 4 different gamma receptors bound to the gamma interferon dimer. And what Juan did was that he selectively ablated the different receptor binding sites on gamma to change the number of receptor occupancy on gamma as well as the location of the receptors. And this is looking at a phospho-STAT1 plot. You see, this kind of looks like the IL-2. We can tile the whole range here of Emaxs for phospho-STAT1 by these partial agonists now. And then what we did was we did some gene expression studies and had kind of a surprising result here, which is that when we looked at gene expression of wild-type gamma versus the partial agonists, we found that most of the genes were activated to a similar degree by wild-type and the partial agonists. But there was a small subset of what we call tunable genes that were activated by wild-type gamma and not that much activated by the attenuated versions. And one of these is PD-L1, and one of the stable genes is MHC1. And so what we were able to show was that when we treat different cell lines with wild-type gamma versus the attenuated gammas, and we look at class 1 versus PD-L1 expression, we see now a bias. And so that's very interesting to me because the possibility that now we can selectively upregulate class 1 in the tumor microenvironment and sensitize T cells to immunotherapy by jacking up tumor antigen expression. Now the bias is different on different cell types. When we look at different cancer cell lines, some of them the bias is quite strong. Others it's very weak. But we're profiling this now and really going forward with the intention of testing the concept that you can sort of turn a cold tumor hot by driving up class 1. Okay, so really the concepts that I've told you about really point to this idea that there's cell type intrinsic response thresholds to cytokines, and that are different on different cell types. And what these partial agonists do is you can essentially shim out different cell types to a given cytokine and narrow the pleiotropy and increase the specificity. For instance, our attenuated IL-10 agonist that Bobby Saxton made in the lab is completely specific for monocytes and doesn't activate anything else. And so we've done this with IL-10, IL-12, IL-2, and many others. And so I think this is kind of a new way of thinking about cytokine therapy and revisiting cytokines in the clinic. And certainly many of the ones that I've talked about today are on a path to the clinic, hopefully. Okay, so what other ways can we manipulate cytokine receptor signaling? So here's another way that we're looking at this. Rather than engineering the cytokine itself, what if we can attack the kinase intracellularly, independent of the ligand binding? So we developed this technology, Ricardo Fernandez in my lab some years ago, called RIPPER, where we make bispecific antibodies that can recruit CD45, and the phosphatase domain of CD45 affects an intracellular dephosphorylation of the intracellular domain. And Ricardo was able to do this to PD-1 and show that we can reduce basal signaling, but we're now able to do this with cytokine receptors as another way of attenuating signaling. And just to show you one unpublished example here, this is a cell line expressing the EPO receptor with the JAK2VF mutant, which is constitutively on and proliferating uncontrollably. And that's what the JAK inhibitors were made for. People with myeloproliferative neoplasm take JAK inhibitors to control those cells. But here's our RIPPER molecule with CD45 recruited to the EPOR, and you can see that it's quite effective at inhibiting proliferation. It's not all the way, but there's certainly an attenuation. And so we're looking at it for these kind of applications. All right, so far I've talked about attenuating cytokine signaling through mutating the dimer interface, RIPPER, but there's another way that I think to me is like the most interesting way is like manipulating dimer geometry. And you heard about that earlier today, one strategy, and I want to tell you about our strategies for doing this. And the way I kind of think about this is GPCRs here again, they function through shape change. And could we take some of that general idea and apply it to dimer structures where we could kind of change the dimer geometry in sort of like a medchem sort of way where we would have molecules that dimerize in different geometries, and then we can select for activity. And maybe we could bias, by changing the geometry, we could bias cytokine signaling in similar ways that the small molecules can bias GPCR signaling. So, Brian Kabilka's in the next lab. He's gotten a lot of attention for GPCRs. I kind of wanted some of that action, and so we kind of went down this path. And so we're collaborating with David Baker with the idea, can we make a rigid dimerizing scaffold that we can systematically tune the geometry? And what we came up with was DARPins, which Andreas Plukten invented and you heard a little bit about today. Well, we made our own DARPin yeast libraries in the lab, and we selected one of these DARPins to the EPO receptor. And here's the crystal structure of the DARPin bound to the EPO receptor. And then with that, I went to David and I said, we want this DARPin to be a dimer and we want it to be rigid. We don't want to just link them together flopping around. And so what they did was they engineered an interface to form a DARPin dimer, a really high affinity dimer. And this is now the structure of a DARPin dimer bringing two EPO receptors together in a completely different geometry than EPO receptor. And one of the interesting things about these DARPins is that they have these little helical repeats that you can insert or delete in them and change the curvature or change the length of the DARPin. And so this is a montage of what we took a DARPin to EPO receptor, dimerized it, and then systematically inserted these little helical repeats to bend the receptor from basically vertical parallel down to horizontal on the membrane. And as you bend the receptor, you kind of phase down the signaling of STAT5 and ERK here, whereas when it's lying flat, it's almost not signaling. And we did the same thing by inserting these repeats in a different way to get a pure translation. So this is twisting as an angle. This is pushing the receptors apart. And again, you can see with different angstrom distances here, it's different here where we have a sort of a systematic phasing out of the signal. Here it reaches a threshold distance and then breaks off, and that's presumably where the jacks just can't reach each other anymore. So we collaborated with Vijay Sankaran at the Broad Institute. He's a hematologist and has a beautiful assay for the differentiation of stem cells to red blood cells, looking at CD71 and CD235, and it has this very nice U shape on a FACS plot over about a 14-day course. And this is what EPO does, starting about here. It sort of comes around to the end of this U here. And when we did this, we placed EPO with our DARPins that enforce these different angles on the EPO receptor dimer. You can see the more straight up angles, you get almost full differentiation, but we can essentially abort differentiation at an early stage by just twisting the receptor down to more extreme angle and arrest these cells in an early developmental stage. And so one of the things I'm really excited about these things is the ability to maybe manipulate cell fate decisions and arrest cells in sort of early fate choices. And so we can't, it's not just twisting angles and distance. We can fuse the DARPins together like a C, and so the receptors go from face to face where they signal to face to back, where twisting the receptor around completely shuts off signaling. So we can play all these games, and it's super interesting, and we're applying them to different receptor systems, but it's like a molecular ruler for structure-activity relationships for receptor dimer geometry. Okay, and so the concept here, again, Junichi talked about earlier, was that dimer matters. Not any dimer will do. And the way you twist the receptor or change its distance can affect the way these jacks see one another and see the substrates that they interact. So with that DARPin experiment, we were really excited. And DARPins, they're in a path towards clinical development, but it's a synthetic molecule. And really, if you want to do this in a way that get people interested in making a drug, you have to do it with an antibody. So we turned to kind of a different strategy for geometric dimer tuning, which is using nanobodies. So here's the IL-2 receptor complex, again, bound to IL-2, has this very stereotyped kind of Y-shaped dimer. We asked, could we just bring these two receptors together with little nanobodies that are very efficient in getting the receptor close together? And the way we get the epitope diversity is that we choose nanobodies that bind to different regions of the receptor extracellular domains. And then we would just link them together. We'd literally fuse them together like beads on a string, and we would do a big matrix of each nanobody against common gamma, against each nanobody with R beta in two different orientations. So this is like what I call cytokine med count. And then we just screened for activity, and we saw this whole diverse spectrum of activities from no signaling to weak signaling to IL-2-like signaling to superagonists that are even stronger than IL-2. And they're not just different in signal strength, they're biased. And so when we look at different representatives of these nanobody dimers, and we characterize STAT5 and AKT, two of the principal downstream signaling axes of IL-2, you can see that you start to get strong biases with some of these molecules where STAT5 is high, but the AKT is low. And that does manifest in functional differences. So when we replace IL-2 with these surrogate agonists in a T-cell differentiation assay, some of them give an effector memory bias, some an effector central, some a central memory bias. And we have other metrics using gene expression really showing, profound differences in gene expression. So essentially what we could have are like multiple flavors of IL-2 that could, each one of them could be potentially useful in a different type of immunotherapeutic modality. And these are just nanobody dimers. And we've determined the structures of some of these complexes, the actual monomer complexes, then modeled the dimer. And you see, compared to IL-2, they're just completely different. This is a superagonist, and it's actually wider apart than IL-2. So again, the role of dimer geometry is just incredibly powerful in tuning cytokine receptor signaling. And I just added this slide earlier today because I just wanted to show you this one other way of making surrogate dimer agonists, which is what we call staplers. It was done by Jamie Spangler in my lab. Whereas if we want to preserve the native dimer structure, use that as a molecular cast to then go search for Fabs that are able to seek out that dimer structure and enforce that on the receptor in the cell membrane. And so I know that this is maybe that's not really clear, but I'll show you a structure that came out of this effort. Here's the structure of an IL-4 receptor, common gamma IL-4 ternary complex, and here's the structure of what we call a stapler antibody, which perfectly, if you look at it from the top, the heavy chain is on on IL-4 or alpha, the light chain is on gamma C. It perfectly bisects this dimer with the CDRs of light chain exclusively on common gamma, CDRs of heavy chain on R alpha, and it perfectly enforces the native receptor dimer of this thing. So this is another way of thinking about surrogate agonists. Okay, so I want to finish up here by going beyond the natural signaling pathways. And one thing I'm really excited about is the idea that kinase receptors at the cell membrane, by virtue of the fact that the intracellular kinases are not absolutely specific for their substrates, that these receptors can be mixed and matched in different combinations to form heterodimers that might have new biology. And we've been exploring this with cytokine receptors. By looking at cells, taking a census of the cytokine receptors, and then creating new heterodimers and asking what they do. And one example is we took a nanobody against the IL-2R beta chain and a nanobody against the IL-10R beta chain, and we fused it together to create a new heterodimer that does not exist in nature. And this molecule, we found 3 combinations that activated STAT5 signaling, and it kind of looks like IL-2. It induces T-cell proliferation to different degrees. It activates ADCC and NK cells to different degrees than IL-2. So essentially, we have like a new flavor of IL-2 that activates STAT5, but through a different kind of heterodimer. So, we're just learning about this. It's like discovering a new orphan cytokine in the genome that has properties that we don't even know what it does at this point. So really, I'm going to finish up here, and just this is kind of the menu of cytokine receptors that are that are expressed on different cells. And to me, when I look at this type of lineup, I sort of think of it as like an artist's color palette, where each one of these is a different color, and sort of nature provided us with the primary colors, but by combining them in different combinations, you can make new hues and tones and other things that may not have been evolutionarily selected because they may not be useful for homeostasis, but they may be really useful drugs. And so we're doing a lot of this scoping around now, looking for different combinations. And then my last slide here, I just wanted to finish up because when we're talking about cell surfacing, receptors, there are just still a lot of orphan receptors out there on the cell surface that we just don't know what they do and we don't know what they're seeing. And that's like a huge problem that has to be solved. There's tons of new biology out there that is remaining to be opened up. And we did a big pairwise interaction screen between all the human Ig superfamily extracellular domains several years ago. 564 by 564, about a 320,000 matrix, using recombinant proteins in a sort of assay, a multiplex assay we set up. And we found hundreds of new hits. One of them that was kind of interesting was we found a binder to PDL2, which wasn't known, called CCAM4. And so when you do these kind of experiments, you wonder like what's junk and what's real. And so to me, the ultimate litmus test of what's real is a crystal structure. And we've managed to crystallize CCAM4 with PDL2. And then so what does it do physiologically? That's a very tough question, but we're starting to look into this. One bit of data I'll show you, we're collaborating with Enfu Hui at UCSD, and we can see that CCAM4 and PDL2 are expressed on the same cell surface on monocytes, and they form cis interactions. You can see here, this is by a FRET assay, cis interactions on cell surface. So we're thinking maybe CCAM4 serves as some sort of decoy on the cell surface to tie up PDL2 and maybe inhibit it from activating PD-1. But this is just the beginning of where we're going with this. So that's all I have to say today, and I think I've called out the people who did this work. It's been a lot of people over the last 5 or 6 years, some names you'll recognize here. These are my disclosures, and I'd be happy to take any questions.
We have time for a couple of questions.
Chris, that is just really beautiful engineering you're doing. And, on so many different types of cytokine interactions, receptor-ligand interactions, developmental pathways, it's really amazing. I'm curious about one point, and that's, I've talked to you before about catch bonds.
Yeah.
And basically, they're the same kinds of things that we work on with integrants, because the integrants, exist in its extended open conformation, which is high affinity, and then they have a low affinity closed state, been closed. And so, when you apply tensile force, you stabilize the high affinity state.
Yep.
And that's true for selectins. It's true for bacterial adhesins that Wendy Thomas works on. But and the problem with the way this has been analyzed in the catch bond field is, like, Qingzhu has never really applied any models of off rates to these measurements.
Yeah.
I mean, we've done it with von Willebrand factor A1 domain binding to platelet glycoprotein 1b, and we can see two different states, and they differ in off rates, and you get. So anyhow, there's a nice physical model for explaining this based on, stable, on the tensile force stabilizing a more extended state, that has higher affinity and a slower off rate. But I just don't see how that comes into play in the TCR
It's a tough one,
MHC interaction.
Tim, I was a skeptic about the catch bonds before. Like doing it, as doing it with your own system will make a believer out of you, and it'll make an evangelist out of you. And
well,
so
it didn't. I collaborated with Qingzhu, and it did not make me into an evangelist.
Yeah. Well, the fact that it's a flat interface is very difficult for me. That's why I showed that sort of cartoon. It's very difficult for me to imagine how a catch bond would be formed. In your initial selectin example, the domain movement, and like we saw in the notch ligands, makes a lot of sense.
Yeah.
But in a flat, so here's how it was explained to me. We collaborated with Bill Goddard at Caltech to do some MD simulations. And he told me, he's a physical chemist, and he said, materials and proteins don't shear like that. They shear like that.
Oh, yeah.
And so my idea, the way I think about this is the TCR shears off the MHC like that, it's picking up adventitious interactions. Just a hydrogen bond or two that just kind of gives a transient little resistance force to hold it up just long enough for that signal to propagate. And that's as much molecular biophysical detail as we have. I agree. It's unsatisfying, but yeah, that's where we are.
Yeah, yeah. All right.
Sure.
We can talk about it later some other time.
Okay. Okay.
Hi, Chris. Thank you for a great talk right here.
Oh, sorry.
It's really fascinating to see how you are using different types of, bifunctional molecules to induce proximity or rigid molecular rulers to change the topology of, cytokine receptors. All the cytokine receptors, they function through a natural dimerization mechanism. So I'm curious to hear your thoughts on the potential of using, for example, rigid modulators to target receptors or membrane proteins that do not naturally function through dimerization, but somehow you could modulate their conformation or geometry.
Well, that's kind of what we did with CD45. We enforced proximity between CD45 and targets that it normally would ignore.
Right.
Right. So, and one thing I didn't show there, the effect is highly tunable. The geometry that you bring CD45 in, the distance you bring it in, will affect the efficiency of dephosphorylation of the target. So,
Yeah.
I think the concept is applicable to a lot of different systems.
Yeah. What about orientation, like modulating things that?
Yes.
Yeah.
Yes.
All right. Thank you.
It matters, yes.
I'll stand on tiptoe. I'm not tall enough. That was great. Absolutely great. I'm curious about the IL-2 and I guess the interferon stories. I think that's just it's so interesting, the effects that you can just dial down the the binding a little bit, and you're hitting these interesting little threshold differences and different stuff. Absolutely fascinating. What do you think's actually is it inside the cell? What's going on? Is it just that it's just not hanging together for long enough for some of the stats to interact, or some of the the phosphorylation to build up?
Yeah, I think
interesting.
I think it's a dwell time issue for the for the transphosphorylation of the JAKs to hit the stats. And so everything is sort of aborted a little bit early.
Yeah.
But so one really interesting finding out of all this is that we thought, well, if we attenuate things, at the membrane, we've done gene expression studies, everything will be sort of correspondingly attenuated to the same degree. But what we find is that some of the IL-2 mutants that have the attenuated signaling, by attenuating negative regulators, we get, increased activity of some of the partial agonists. Right? Because we're not activating the negative regulators as much.
Yeah. So they're getting a bigger piece of the cake.
Yes.
I think it's really nice.
Thank you.
Really, really interesting.
Thank you.
Hi, Chris. Fascinating talk. So we study inflammasomes, and so the cytokine signaling is really fascinating. So I meant to ask you about the interferon gamma signaling story you just mentioned. So I think you mentioned that when you engineer the interferon gamma, you can change how many receptors engage, therefore change the expression of PD-L1 in some of the cell types. Did I catch it correct?
Yeah. So in the signal attenuation, there's a greater reduction of PD-L1 than there is class 1.
So what is, I guess, and it appears to be different for different cell types.
Yes.
Is that right? So
Yes.
that seems to imply that in different cells, you actually have other receptors or adapters or whatever partner proteins that engage the receptor in a different way.
No, I don't think that. I think it's just that, I think in different cell types, it goes back to this hardwiring issue. I think cytokines activate a lot of genes, and I think they come on early and late, and some of them are sluggish, and some of them are like, for gamma interferon, class 1 is like why it exists, right? And so it's like the first thing to come on. You need like a molecule of gamma interferon to jack up class 1 to the max. And I think other molecules like PDL2 are not as germane sort of to gamma interferon's main function. So maybe they're just later to come on. That's how I think about it.
that may have to do with the expression level of those two
Yeah, it could
components
very,
of different cell types.
could be, yeah.
Right. So
yeah.
I have a follow-up comment on the
Yeah.
catch bond that Tim was asking about. So you were mentioning about this alanine, was it 98 or 92
Yes.
or 98
Yeah.
in TCR? And you mutated to, I noticed you mutated to aspartate or lysine, and you have different effects. So I wonder if that alanine is in contact with its partner in some way.
No,
That forms a catch bond? that you
It's,
can
it's
mutate
actually in the
otherwise?
structure. And so for our catch bond engineering, we don't touch amino acids on the TCR that are in direct contact with the MHC
I
or
see.
the peptide. We want them a little further away. So we're not just affinity maturing the interaction.
What I meant was, is that alanine in contact as you slide?
Oh, I don't know. I mean, that's a dynamic process. We just can't see that. You know, this is the frustrating thing about getting more molecular clarity about this process. It's a transient state.
I see.
Yep.
One final question, please.
Really, really interesting work. I'm just wondering if you have any thoughts on, in a therapeutic setting, if you wanted to activate a subset of cells to treat an indication in which that subset may not be acting properly, could you, rather than having the therapeutic be made of an engineered cell, potentially use these mix-match receptors as the therapeutic itself to fix or just activate, recruit the cells to the site of injury or disease?
Yeah, you know, I think conceptually that's what we're trying to do. We're trying to, you know, right now in immunotherapy, there's the synthetic cell approach and the protein approach. And so what I think is fun is trying to mimic what you can do with an edited T cell with a recombinant protein. So I don't know what specific indication you're thinking of, but that's kind of what we're trying to do. Okay. Okay, thanks.
Thank you.